So we talked about the structure of enzymes, but now it's time to get a little bit more advanced with this and talk about substrate fit. And what I mean by substrate fit is its ability to fit at the active site of an enzyme. And this is based on two things. The first is the shape of the substrate, and the second is the charges involved. It's not necessarily going to be a full charge. Another important way that substrates interact with enzymes is through partial charges. We're going to walk through these individually, so let's start with shape. What I'm doing here is putting in two enzymes with substrates that look as though they will fit like a puzzle piece into the enzyme. And that's exactly what happens. I labeled my enzyme with E and my substrates with S. One thing that's really important for us to note, it does not have to be an exact fit for the enzyme to function. So here I'm drawing a very squiggly looking active site for my enzyme, but I'm keeping the substrate the same. That nice, neat triangle. And here I'm showing you how that substrate will come into the enzyme's active site. And what happens here is that the active site then molds to the substrate. It changes its conformation, another word for shape, so the enzyme changes its shape to mold to the substrate. So if we had a giant square, yes, that would not fit. However, this enzyme is able to mold to the triangle that we have. And this is what we call induced fit. This contrasts the old model that we used to think was the right one, and that was a lock and key model. We now say that we agree with an induced fit model. So now we're going to talk about the second component here. And remember, the second component of substrate fit was charge. So what I'm drawing here is an enzyme and a substrate. And the substrate goes on its business in the cell. There's a lot of random molecular motion, and maybe it hits the enzyme, but then it will probably keep going on its merry way. Things in the cell are constantly in motion, nothing static. So we can hit the active site without sticking to it. So that's where charge comes in. Because the charge differences allow for the enzyme to hold on to the substrate. And remember that this occurs at the enzyme's active site. And when I'm saying charge differences, what I mean by that is opposite charges, because remember we said that opposites attract. So I'm going to draw a very simplified version of this enzyme and the substrate. And I'm going to put charges on it. So at the top of the substrate, I'm going to have all positive charges. And then at the bottom, I'm going to say positive, negative, positive. These differences in charge are partial, meaning they're caused by hydrophobic effect or by hydrogen bonding. And then there's going to be the opposite charges on the enzyme, so negatives on the top, and then negative, positive, negative on the bottom. So now random molecular motion is happening, and this enzyme is just sitting there. This substrate makes contact, but now because of the opposite charges, it's going to be stuck in there. And I know you guys can't animate it like I just did, so what you can do is just draw that arrow in. And the result that we get is this substrate stuck in this enzyme with the opposite charges attracting. So now that enzyme in the active site is able to hold on to that substrate. These opposite charges are not full charges. Instead, they're partial charges. This is important because if it was a full charge, that substrate would never be released by the enzyme. But we can say that the partial differences in charge allow for induced fit to take place. We know that the substrate interacts with the enzyme at the active site, but so what? Why do we care? We're going to take an example to work this out. Our enzyme in this example is going to be called lactase, which is a very, very, very helpful enzyme because you need to break down lactose. So if someone is lactose intolerant, that means they don't have enough of the lactase enzyme. That also means you're in the club with me. But lactose, remember OSE means it's a sugar, and to break down lactose, living things need the enzyme lactase. So we have our enzyme lactase, we have our substrate lactose. And this substrate lactose cannot be digested or absorbed by our cells as is. Okay? So I'm going to make a very, very simplified drawing. We're going to say this is lactase. And this is our substrate, lactose. If you remember our discussions towards the beginning of the year, it's important that we understand our sketches as models. In real life, the lactose here would be much smaller than I have drawn it, probably the size of the O in lactose. Highlighting the active site. What will happen is when this lactose, it's going around, it has random molecular motion, just doing its own business, and then boom, it finds itself in a lactase enzyme. Once that lactase gets a hold of our lactose, we can make a drawing that looks a little bit like this. Right, so our lactase is hanging on to our lactose. When it's in this conformation or this shape, we call this the enzyme substrate complex. We need to break down that lactose. This last piece is going to be step 3. And what's happening here is that the enzyme is wiggling ever so slightly with that lactose inside it. So the lactose is coming in, sitting in here, and then the enzyme is just going to have molecular motion as enzymes do. It's going to be moving around. At a certain point, it's going to be moving around too much for that lactose to stay in one piece. Remember, there are partial charges on the substrate that we talked about earlier. These are opposite to the partial charges on the lactase enzyme. When the enzyme stretches, the two sides of the lactose molecule are pulled apart from one another. This breaks a chemical bond in the lactose sugar. And now we have lactose broken into two pieces. And then these leave the enzyme. Just draw them like this. So you know there's two halves. Used to be lactose, but now we have glucose and galactose. These we call our products. And luckily for us, these can be digested or absorbed by our cells. So we have step 1, the lactose moving around in the cell. Eventually it's going to hit a lactase at the active site. Then we have step 2, which is it's stuck in that active site. There's some charge interactions going on between the substrate and the enzyme. And then we get some motion here, and eventually our substrate is broken. Here we broke lactose down into glucose and galactose. Those are our products.